Good day, everyone. On behalf of Cambridge Health Tech Institute's Global Web Symposia Series and our sponsor, Metabolon, I'd like to welcome you to how metabolomics is uncovering a greater understanding of cardiometabolic disease. My name is Elizabeth Lamb, and I'll be the host and the moderator for today's event. Now I'd like to introduce our presenter for today. He is Dr. Martin Hornshaw, PhD, Director of Scientific Marketing for Metabolon. Welcome, Dr. Hornshaw. The presenter ball is yours.
Thank you, Elizabeth. And thanks, everyone, for joining. I look forward to spending the next 55 minutes or so with you and telling you about metabolomics and how we're uncovering a greater understanding of cardiometabolic diseases. Before we actually get started, I heard a piece of news today. I live in the UK, and this was on the BBC. And they were telling us about a meta-analysis of dozens of papers published in the Journal of Molecular Psychiatry. And it was relating to mental health. And it was looking at the effect of diet on the development of depression, which I found quite interesting. It noted that a Mediterranean-style diet rich in plant material had an inverse association with depression. So you were less likely to be depressed if you had a Mediterranean-style diet. And it noted that this seemed to be mediated via things like inflammation and the microbiome, the gut microbiome. And it put me in mind of my talk today because these issues of diet, of your microbiome in the gut, dysbiosis, etc., they can affect areas of health that might be unexpected, such as whether or not you develop depression and whether or not you develop a cardiometabolic disease. So I just thought I'd mention that. You might want to look it up in the Journal of Molecular Psychiatry. So, why is metabolomics useful? What is it about metabolomics that helps you understand biology, that helps you understand disease? Well, I'm sure you're well aware, you've been to a physician in your life, that metabolites are diagnostic. Things like glucose, cholesterol, triglycerides, steroids are already used as markers of health. And the reason that metabolic homeostasis is relevant is that it's interlocked with physiological processes. They're coordinated. So an alteration in one will affect the other. And metabolites offer for us a way to integrate together the information that we need to understand complex traits, such as diabetes, such as obesity. And the inputs to developing that are obviously coming from genetics, coming from the microbiome, coming from our environment, coming from our diet, coming from stochastic events like infection, like accident. So metabolomics, if you remember the basic paradigm of biology, and you do, genome, RNA, protein, the next step is metabolites. Metabolites are, we would argue, the closest thing you can get to phenotype, and they are in themselves an intermediate phenotype. The kind of metabolomics that we perform, we call it Global Metabolomics. Its goal is not to analyze an individual metabolite one at a time or in small panels. The whole point of it is to look at huge swathes of metabolism at the same time, and by doing so, look for disease-related changes that could be unexpected to provide a snapshot of health. So what that means is if you measure metabolites, you can assess and understand the physiological state. And if you measure many metabolites with a global metabolomics approach, such as by Metabolon, you get a broad understanding of biological change, which can result in disease. So I was reading a paper a few months ago from Bommer et al., and it's in Lancet Diabetes and Endocrinology, and it's set out to estimate the global cost or burden of diabetes on the economy in 2015. Of course, we can have an individual cost, which is very much important to our health, but there's also that global economic cost. And I was staggered or even taken aback. At the same time, I was, for some other reason, looking at the GDP of certain countries, and I noticed the estimated global cost of diabetes in 2015 was $1.3 trillion. And that's very similar in size to the entire economy of Canada. So perhaps it's a strange analogy, but if diabetes was a nation state, it's so big that it would be in the G20. Okay, so let's discuss in the next few slides how Metabolon performs metabolomics, what kind of information you can obtain from it, and then we'll get into some examples. We've been doing this for quite a long time now, since 2000, and we've had experience with several hundred different types of matrix. So what we want our clients to do is to send us their samples, and then we extract the metabolites. We put them through our liquid chromatography mass spectrometry platform, and you see there are 4 branches, 4 arms to that platform, because there are small metabolites, big metabolites, hydrophobic, hydrophilic, etc. They have different physicochemical properties, and one LCMS method cannot identify them all or quantify them all. What happens if you simply inject water into that LCMS platform for metabolomics, our precision metabolomics platform? You see thousands and thousands and thousands and thousands of peaks. Incredibly pure water, because mass spectrometry is so incredibly sensitive, it's still possible to detect huge numbers of really, really trace compounds in the water. So you see those coming from pure solvents. They still have trace contaminants in them, and you see them coming from the air. So when I used to work in the lab, I don't anymore, you would walk into the lab in the morning and you would get strong background signals coming from the operator's deodorant, and they would fade during the course of the day. So there's thousands and thousands of real signals coming from the water, from the solvents, from the air. So what you need is a way to remove all that background, which is actually noise, but is in essence background, it's useless information, and then reduce down the data to the functionally useful data. A single metabolite could give you 10 or 20 signals of dimers or fragments of metabolites that are bound to sodium ions or something like that. So we've got a long, long history of doing this, and we know how to do that. We have a bioinformatics platform which enables that. The next thing to do is to identify the metabolites, and we've gone to the extreme trouble of creating an enormous synthetic, authentic chemical standard library. All of these thousands of chemicals and biochemicals have gone through our precision metabolomics platform, and we've acquired information about retention time index, about the accurate mass measurement, and about MSMS, which allows us to then compare to the sample. So we can identify categorically, absolutely, this is such and such a metabolite, not a putative identification. So we like to think that we're a biology knowledge generating machine. So the samples come in, they're logged, they're processed, they go on the LCMS platform, they come off, they go through multiple rounds of quality control, then they go to statistical analysis, and we'll discuss with our clients what analysis that should be, and then they go to a biochemist. And that biochemist owns the return of data to you. So they're all XPIs or people from the pharmaceutical industry. So what they will return to you is an Excel file with a list of metabolites, the super pathway, the sub pathway, links to various pieces of public information, heat maps, and so on. But what they also do in addition to that is interpret the data. Well, at the start of this year, Metabolon had run 7,000 studies. At the end of this year, we'll have run about 8,000 studies. And that's given us, as well as published information, a huge institutional knowledge in the area of metabolism. And because of that, we think we're very good at identifying biomarkers associated with disease or some phenotype, about delivering mechanistic understanding in biology, about uncovering the mode of action of a drug, for example. So if you do that, you'll identify in a sample like plasma about 1,000 homeostatically maintained metabolites. Now, if you also run a lipidomics platform here, you'll identify 1,000, 1,100 actually, complex lipids that are also homeostatically maintained in plasma, so a total of more than 2,000 molecules. Complex lipids, I mean things like ceramides, DAGs, tags, various phospholipids, and so on. And as I've already mentioned, but I'll reiterate, when you combine the genomic information with external factors like diet, exercise, medications, etc., microbiome, that produces phenotype. And because metabolomics is so close to phenotype, it gives you a snapshot of what's going on in the biology at that instant in time and can help you elucidate health. What you're seeing here, this is a piece of metabolism from Metabolon's proprietary map of metabolism. It's a little bit different to the published maps of metabolism. And what you're viewing is statistically significant changes in metabolism for a patient. So the red and blue dots and the size of the bubbles indicate the change in metabolism, more of that metabolite, less of that metabolite, and the amount of change. We're only showing you the statistically significant ones in this instance, not all the metabolites. So what you're viewing here is the data from the patient, and this is a collaboration we do with the Baylor College of Medicine in Texas at their Department of Human Genetics, and they asked Metabolon to help them identify inborn areas of metabolism or inherited metabolic disorders. I think your eye, even without the oblate circle, would be drawn to the left-hand corner where you see lots of red bubbles of different sizes and lots of blue bubbles of different sizes. And you can almost imagine a line in between them where there's some kind of break in the metabolism. Above, you've got excess amounts of metabolites compared with normal, and below, you've got reduced amounts of metabolites compared with normal. What you're viewing here is a sample which is taken from a child who's suffering from a disease, and we've identified it as maple syrup urine disease. And that's caused by a mutation in branched chain keto acid dehydrogenase. And now we've had enough experience with Baylor College of Medicine, we've been able to map out the profiles of the altered metabolites that indicate dozens and dozens of these diseases. Because we've got this enormous library and this enormous history of doing metabolomics, we see all the super pathways and sub-pathways that is possible to see in a method which takes a total of 7 minutes. So when we analyze a sample, it takes 7 minutes, and you automatically get the data coming off the mass spec. So we see amino acid metabolism, carbohydrate, nucleotide, etc. But as well as all those mammalian metabolites, we also see novel metabolites, such as metabolites of drugs of abuse, xenobiotics, such as plant metabolites from the diet, microbial metabolites. We see hundreds of those in our library. So we offer a really comprehensive approach to analysis of metabolism, both mammalian, microbial, and external sources of metabolites like the diet. And I just spent a little bit of time on the microbiome, and then we'll start looking at some examples of the biology that we hope we've helped our clients with. So you're seeing here some examples of microbially derived metabolites, or metabolites that come from bacteria and eukaryotes. And for example, I'll draw your attention to secondary bile acids over here. And on the right, they are lipid signaling molecules, and for example, they can tell you about liver health and liver disease. In addition to these bile acids, you also have, for example, neurotransmitters. A lot of neurotransmitters are created by microbes in the gut, and obviously they directly affect the brain. But some neurotransmitters, such as serotonin, their levels are modified by dysbiosis in the gut. Okay, let's talk about our first example. This one was published in 2016, and it's by Theis et al. From the Weizmann Institute, and it's published in Nature. And it's trying to elucidate mechanisms which drive the yo-yo dieting effect, where someone is perhaps obese, loses weight on a healthy diet, they go back to what they would consider a more normal diet, and then they gain weight again, and so it goes. This one resonates very well with me because I've suffered that in the past. So we want to elucidate mechanisms driving this effect, and the study paradigm was to look at a mouse model fed normal chow, a high-fat diet, or something in between, or one of those in-between things was a cycling high-fat diet, measure weight gain, measure the gut microbiome, and also perform global metabolomics. Now, what you can see over here is these are 3 measures of the diversity of the microbiome, and I'll draw your attention to one of them, particularly alpha diversity. So you see here in black, mice fed a normal chow, then in red, before obesity, and blue with a high-fat diet, cycling high-fat diet in red, and in blue a high-fat diet. And measured before obesity, during obesity, you see it drops in the floor, particularly for those on a high-fat diet and a cycling high-fat diet. And it recovers a little. Measures of OTUs and genes tell you the same story. There are significant changes on a high-fat diet and a cycling high-fat diet for mice in their microbiome, some of which is never recovered. Now we see a similar picture with the metabolome. In the green box here on the right, you see examples of metabolites which have dropped significantly or are absent now because of a high-fat diet during obesity and after obesity. And I'll speak to a couple of those, apigenin and naringenin now. So they're examples of flavonoids from plant material, from a plant diet, which you find in a normal chow over here, but you see much less of in a high-fat diet. If you look at the top left chart, before obesity, you see apigenin is high, it kind of tanks during obesity, and it barely recovers even after 15 weeks of obesity. 15 weeks is quite a large amount of a mouse's lifespan. And the same picture for naringenin. So the high-fat diet contains less apigenin and naringenin, but also, I should point out that this is a logarithmic scale. Before obesity, during obesity, after obesity, you have a kind of inverse of this picture. This enzyme, flavonone 4 reductase, is massively overexpressed by the bacteria in the gut, as obviously they're scavenging to take those flavonoids and utilize them. They're obviously important for bacterial metabolism. Let's look at the actual effect of those different regimes of normal chow and high-fat diet on weight. And then what does that mean? Well, in black, you see normal chow, there's a slow weight gain over those 20 weeks. Now, the blue line represents the mice fed high-fat diet, and they gain something like 70 or 80% weight compared with the mice fed only normal chow. They really put on a huge amount of weight. If you look at the green line, that's the mouse that encounters only one round of high-fat diet. It gains weight like normal chow fed mice until it encounters a high-fat diet. It gains weight, then it loses some. It doesn't quite return to the normal chow level. Looking at the red line, that's a mouse that's primed with a high-fat diet, then normal chow, and then it encounters a high-fat diet again, simulating a yo-yo obesity effect. It gains weight, loses weight, gains weight, even more weight, and then it loses weight and ends up somewhere intermediate between normal chow and a high-fat diet. Now look at what you do if you supplement the mouse's diet and the second round of high-fat with those flavonoids, apigenin and naringenin. It tracks with the red line, then it tracks with the green line. And then it returns to a weight as if it was fed normal chow only. So in effect, that flavonoid supplementation treated and cured yo-yo obesity. Why is that? Well, this is just a control over here on the right, the top chart, to show that in the orange mice, the mice with supplemented flavonoid, those flavonoids return to normal. The biology underpinning this, comparing the red with the orange, the without flavonoids in red with the plus flavonoids in orange, and looking here at the axis, you see that the mice are burning more calories. They're increasing their energy expenditure when they have flavonoids in their diet. And just briefly, that relates to brown adipose tissue and heat generation by uncoupling protein. So there's a mechanism why the mice with flavonoids are able to beat the yo-yo dieting effect. So just to summarize, there's a major reduction in microbiome diversity after cycles of high-fat diet and doesn't recover even after 15 weeks. The same is true of the metabolome. I didn't talk about this, but fecal transfer experiments showed that accelerated weight regain phenotype with the high-fat diet cycling can be transmitted to germ-free mice. Reduced levels of flavonoids in post-dieting mice contribute to a reduction in energy expenditure, which exacerbates weight regain. And flavonoid supplementation therapeutically ameliorates the rate of secondary weight regain, such that the weight of the yo-yo mice supplemented with these flavonoids, which we uncovered with the metabolomics approach, return to the same weight as those fed normal chow. In my next example, I'm going to carry on talking about obesity, but also blood pressure. This is a study in UK twins of about 2,400 women. What was being studied was what metabolites associate with those three things: blood pressure, visceral fat, insulin resistance, and type 2 diabetes, and which metabolites associate with both. So for example, over here in the blood pressure part of the Venn diagram, you see that these are dicarboxylic acids, are fatty acids that have a carboxylic acid group at either end. They associate only with blood pressure. And elevated levels, for example, of hexadecandioate have been shown to be associated with blood pressure, hypertension, and to be causally associated with heart failure. And I'm going to explain that in the next slide. But let's look at this intersection between insulin resistance and visceral fat. You see these metabolites here. All of these metabolites relate to energy metabolism. There's branched chain keto acids, branched chain amino acids, glucose, and lactate. And they're associated with the citric acid cycle and glycolysis. On average, 39% of the association of these metabolites with insulin resistance was mediated went via visceral fat. So there's a large contribution of visceral fat on insulin resistance related to these metabolites. Now, the authors hypothesized that reductions in visceral fat mass may potentially result in improving mitochondrial TCA or citric acid cycle efficiency seen in patients with diabetes. And it was consistent with a recent therapeutic trial which treated diabetics with a very, very low calorie-restricted diet. And in 50% of those diabetic patients, they recovered. They returned to fasting glucose levels. So the last thing I'm going to mention now is that there's also something similar going on between blood pressure and visceral fat. There's also energy metabolism altered, but a different kind of energy metabolism, this fatty acid palmitate. Other things that are different between visceral fat and insulin resistance and visceral fat and blood pressure are this peptide. It's a marker of inflammation. So we have overlapping but fundamentally different biology going on in the impact of visceral fat on blood pressure and the impact of visceral fat on insulin resistance. So visceral fat mediates some of the risk of insulin resistance and blood pressure, although in different ways, and it's linked to these metabolites. I mentioned hexadecandiolate. So there's several papers here, so this is a complex slide. I've added notes. I hope that everyone will find them useful if they want to view this at a later date. So this is quite a few studies. It was a study in people of African descent and people of European descent and in animal models, where it looked for associations of metabolites to rare gene variants and if they would then go on to impact blood pressure, hypertension, and heart failure. And what I identified using an approach called Mendelian randomization, and I'll explain that in a moment, is that this rare variant of SLC01B1 was associated with elevated levels of this dicarboxylic acid, hexadecandiolate. If you look here, you see one loss of function mutation increases the amount of hexadecandiolate. Two of them increases the amount of hexadecandiolate. And that increased the risk for heart failure by 29%. Now, Mendelian randomization is a way of assessing causality in a population study. So the way it works, or the basic fundamental principle of it, is that during conception, our genes from our two parents become randomized. And this allows us to overcome confounding factors. So there's kind of three parameters that you look at. You look at a genetic instrument, which is the gene, the variant, and you look at its association with an outcome, a phenotype, in this case blood pressure and heart failure, and what mediates that. And what mediates that here is a metabolite, so we're looking at the levels of metabolites compared with variants. You can look at a forward association and a reverse association. So does that increased level of hexadecandiolate lead to disease, or does that disease lead to increased levels of hexadecandiolate? And it's only the forward association. The hexadecandiolate leads to disease. Now, if you're looking at a translational animal model, a rat here, this is systolic blood pressure against time. When you give a group of mice hexadecandiolate in blue, you see their systolic blood pressure increases. So you've got a very interesting and convincing story that in more than one cohort, people from different backgrounds, racial backgrounds, and in an animal model, hexadecandiolate increases blood pressure. Okay, let's look at the third paper, Many et al. And PLOS One. Now we'll look at this chart, which depicts the three steps of omega oxidation of fatty acids to produce dicarboxylic acids. So in a different cohort of UK twins, a subsequent GWAS study, which also looked at the transcriptome levels in adipose tissue, identified that alcohol has the strongest effect on individuals with high levels of hexadecandiolate. I mentioned that hexadecandiolate is a product of this omega oxidation, which is a secondary fatty acid oxidation pathway. The enzymes involved in this pathway are associated with this phenomenon of high levels of hexadecandiolate being affected strongly by alcohol. So CYP4, ADH1A, ADH1B, mRNA levels in adipose tissue correlated with high hexadecandiolate levels, which regulates the effect of alcohol on blood pressure. Now, this just tells us something significant about a potential future change in clinical care for people who have hypertension caused by alcohol. So the strategy for reducing blood pressure in such alcohol-induced hypertension could well be different depending on whether a subject has a high hexadecandiolate level or a low hexadecandiolate level. And a therapy might be different yet again if metabolites indicate that high blood pressure is associated with the visceral fat. Okay, let's look at the impact of diet on cardiometabolic health. I've got a number of these examples I'm going to take you through where we use metabolomics to understand something fundamental in biology relating to cardiometabolic health. Now, this was a population study in 2,000 female twins. The twins were given food frequency questionnaires and asked about their diet. Then metabolomics was performed and various measurements were made of obesity and for cardiometabolic markers such as inflammatory markers, insulin resistance, dyslipidemia, hypertension, metabolic syndrome, over here on the right. And they were associated with twins who had a high intake of branched chain amino acid in the diet or a low, and it was split into quintiles. So you compared the highest to the lowest quintile. The highest quintile of twins who the quintile of twins with the highest level of branched chain amino acid intake had the lowest levels of insulin resistance, lowest levels of inflammation, and the lowest levels of hypertension. They also had the reduced weight, they were less likely to be overweight. But when you looked at discordant twin pairs specifically, so a pair of twins, one with high BCAA, one with low BCAA, this disappeared. Other things that the metabolomics uncovered were metabolites which had an inverse association with BCAA intake. So you've got biomarkers, alpha-hydroxy isovalerate, trans-4-hydroxyproline. We also looked at the branched chain amino acid levels in the blood, in the circulation, and that was not associated with these biomarkers. It was only the branched chain amino acids in the diet. So these findings, again, lead to a conclusion that might affect health. Branched chain amino acids may be cardioprotective, and this study demonstrates the potential for improvements to public metabolic health by modification of the diet to increase BCAA, branched chain amino acid intake. And I should point out that it's eminently achievable. The difference between the lowest and the highest quintile of BCAA intake was the equivalent to a glass of milk. So there's an old advertising slogan in Britain from when I was a child, drink a pint of milk a day. And American friends and colleagues might remember that slogan, got milk. It seems to be good advice. So we can also understand mechanism of action of a drug. So in this slide, we're looking at the mechanism of action of metformin, and it's studied in a cohort of German people. They're called the Cora F4. And they were grouped according to whether they had normal glucose tolerance, they were pre-diabetic, whether they were diabetic without metformin treatment, or whether they were diabetic with metformin treatment. And the red diamond is indicating the level of citrulline. And what you see is metformin treatment is knocking down the level of citrulline. This blue cross was a putatively identified metabolite. It's one of our what we call X-metabolites. We haven't synthesized the chemical and identified it categorically. So we thought this was a metabolite called 5-trimethylaminovalerate, which is a good microbiome metabolite but measured in the circulation. So that's going up when you have metformin, and the citrulline is going down. I should point out that the authors also looked at an animal model, a mouse model of diabetes, and they took 4 different tissues to see what the effect of metformin was on the metabolome, and they saw similar effects in plasma, skeletal muscle, and epididymal adipose tissue, but not in liver, of citrulline and these other metabolites, but they didn't see the same thing for the microbial metabolite. So in a sense, the mouse model was not a perfect model. It showed the mammalian metabolism to be the same as the human, but the microbiome metabolism was different. So let's talk about the biology and what seems to be happening. So patients taking metformin, they have an upregulation of AMPK, that's 5'-AMP activated protein kinase, and AMPK plays a role in cellular energy homeostasis and largely activates glucose and fatty acid uptake and oxidation when cellular energy is low. Now, in turn, the AMPK in the liver is increasing endothelial nitric oxide synthase. Now, that affects the nitric oxide cycle, which is interlocked with the urea cycle. So you're seeing reduced levels of these urea metabolites, citrulline, ornithine, urea, and arginine, which are interlocked also with the nitric oxide cycle. So we can see that the effect of metformin is ultimately to turn down the urea cycle. I mentioned that X metabolite, and I thought I'd take a moment just to tell you how you go about identifying it and giving an example. So at the time we published that, Adam et al., or rather we produced the work that was published in 2016, that was still an unknown metabolite with a putative identification. In 2018, we co-authored a paper where we published the identity of that metabolite, and it was as we had suspected. And that was in a study of a cohort of Qatari people. So if you look at the data, what you see here is an extracted ion chromatogram from the chromatography of the, on the right of the unknown metabolite, and underlying it very beautifully is the candidate molecule. It has the same mass within the limits of the mass error of the mass spec, 160.1333, and the mass spectra, again, within the limit of the performance of the mass spec, they're essentially identical. So we have a very strong identification that that unknown metabolite was indeed 5-trimethylaminovalerate. Sorry, slides is not my strong point. This isn't a great slide, but let's speak to it. This is a very recent study of, again, middle-aged women studying the impact of the microbiome and the metabolome on arterial stiffness. So major adverse cardiovascular events are not so rare, and we know the causes of some of them, but not all of them. A large number of these major adverse cardiovascular events are not explained by traditional cardiovascular risk factors. And one suspected risk factor is arterial stiffness. And the gut microbiome has been implicated in a variety of potential disease mechanisms, including oxidative stress and inflammation, that could, of course, influence vascular disease. So many et al. Wanted to look at the potential impact of microbiome dysbiosis on increasing arterial stiffness in women and therefore increasing the risk of a major adverse cardiovascular event. So in these 600-odd women, they took a large number of measurements of blood pressure, the carotid femoral pulse wave velocity, this is the gold standard measurement of arterial stiffness. They measured visceral fat mass, fasting insulin levels, calculated the risk of atherosclerosis, took a food frequency questionnaire, what are you eating? They looked at inflammation, they measured the microbiome in the gut and feces, and they looked at serum microbial metabolites. So they took a lot of phenotypic measurements. The upshot was that the proportion of variation in arterial stiffness, so I'll repeat that, the proportion of variation in arterial stiffness that could be explained by visceral fat and insulin resistance was very small, it was 1.8%. But the proportion of arterial stiffness in this study explained by gut microbiome diversity, individual species of microbes, and microbiome-derived metabolites, phenyl acetylglutamine and indole-propionate, was much higher. So, in terms of arterial stiffness and increasing the risk of major adverse cardiovascular events, this opens up a therapeutic route. Adjust the levels of the metabolites, probiotic supplementation, alter diet, targeting the gut microbiome to reduce the risk of arterial stiffness. Okay, this was a paper published again in 2018, quite a significant paper in my opinion, published in Nature Genetics, and it looks at the heritability of the microbiome. How heritable is the microbiome, and what's its influence on health, and what's the mediation on the metabolome, the fecal metabolome. So the heritability of the microbiome per se was only 22%. And the heritability of the fecal metabolome was even less, quite modest, at 17.9%. So they're not highly heritable characteristics. Now, when you looked at the gut microbial composition and associated it with the metabolism, it explained 2/3 of the variation, observed variance, of most of the measurable metabolome, of 710 metabolites. I'll repeat that. The gut microbial composition explained most of the observed variance in the levels and the amounts of those 710 metabolites. 102 of them were associated with visceral fat mass, and only 8 with BMI. Of those 102, 43 were amino acid metabolites. So it's highly likely that microbial amino acid metabolism is centrally involved in central obesity. There were other strong associations. For example, B vitamins and other antibiotics. Their association was 100%. There was no heritability whatsoever. And they showed extremely strong associations with microbial composition. And so it went. There's a very great deal of association. Hundreds of metabolites were associated with hundreds of OTUs, and many of them specifically at the level of the taxonomic unit, not at the higher level. So there's a high degree of interrelatedness between the gut microbiome and the fecal metabolome. So if you like, the fecal metabolome tells you about the gut microbiome and vice versa. Since diet is modifiable and affects microbiome composition, but our genomes are not modifiable and they're not really having a great deal of effect on the microbiome anyway in terms of heritability, this offers an avenue to understand the reasons underlying the development of obesity, how to modify it to promote health. Okay, I've got a couple more examples, and then I'm going to talk to you about a bigger picture of using metabolomics and other omes to address how we can maintain health rather than treat disease. So let's look at this paper from Price et al. In Nature Biotechnology last year. And the leader of this team was Lee Hood, who you could think of as the father of systems biology and the father of systems medicine. And I'll speak to this paper again later in the health section. But let's look at it from the point of view of a cardiometabolic marker. So what they did was they took multiple measurements, 3 measurements of the metabolome over time for 108 healthy individuals. They also measured the genome, proteome, and the microbiomes. They also took more than 200 clinical laboratory tests, and they looked for the association of the various molecular markers, proteins, metabolites, etc., with clinical labs. And the metabolomics had an enormous correlation with the clinical labs, as you see on the left in A. That's the first major finding. Another major finding was that they could, looking at the clinical laboratory tests, these are clinical laboratory tests that measure some aspect of cardiometabolic health, what molecules associated with these clinical laboratory tests. And what you're seeing here is a Spearman correlation, and everything here is statistically significant. So in purple, these are metabolites. In red and orange, these are clinical laboratory tests. And these others here are the other types of molecule, proteins and microbiome, for example. So there's an enormous correlation of metabolites measured by metabolomics with these clinical laboratory tests. And you can see these purple and red lines really funneling down to one point. And this is a metabolite called gamma-glutamyl tyrosine. We're showing here some of the examples of statistical significance in association with markers of cardiometabolic health, such as small LDL particle number, insulin, glucose, HOMA-IR, triglyceride levels, and HDL cholesterol. What this paper seems to have uncovered in just a study of 108 people is that gamma-glutamyl tyrosine could be a candidate biomarker for diabetes risk completely independent of BMI. So this paper is so hot off the press it hasn't even been published yet, or it hadn't been when I checked a couple of weeks ago. It's in the Bioarchive for people to look at. And it talks about a profound perturbation of the human metabolome by obesity. So it took anthropomorphic and metabolic measurements at 3 time points over a median of 13 years and about 2,000 twins. The largest time difference between the first and last measurement was 18 years, which again is a huge proportion of an adult's lifespan. It also looked at a single time point for 400 or so completely unrelated volunteers. What they identified was that the great majority of people of a normal weight had a consistent metabolome, and the great majority of people of an obese weight had something which is characteristic of obesity. It was an obese metabolome, and they had an R-squared of something like 0.9. But there were outliers. There were people of a normal weight which seemed to have the obese metabolome, and there were people of obese weight which seemed to have the healthy metabolome. So because they've taken data over so many years and followed these people, they can look at health outcomes. And what they found was that an abnormal metabolome signature, which indicated an obese metabolome, irrespective of BMI, was associated with a 3-fold increase in cardiovascular events, and it also associated with an increased risk of insulin resistance. I find that a very significant finding. So in the last few minutes, I'm going to talk to you about some genomics and metabolomic studies of healthy individuals. Can those studies provide actionable data to maintain or improve health in these nominally healthy individuals? So first, this pilot study, which looked at whole genome sequencing data in primary care of nominally healthy individuals. And basically it found that whole genome sequencing data was not sufficient to assist the healthy in remaining healthy, and I'll just read this out. A recent report of a pilot randomized trial of the utility of genomics applied to the healthy did not support the use of whole genome sequencing in primary care. Adding whole genome sequencing to primary care reveals new molecular findings, but they were of uncertain clinical utility. So let's move on to some other studies, one of which I've already mentioned, Price et al., but I'll go into more detail about, which looked at combining genomics with metabolomics, and for a couple of studies, other big data as well. And from this paper, Price et al. From the group of Leroy Hood, the defining feature of systems medicine is the collection of diverse, longitudinal data for each individual. These data sets can be used to unravel the complexity of human biology and disease by assessing both genetic and environmental determinants of health and their interactions. Let's look at one of the earliest studies of the combining genomics data with metabolomics, and it's published in 2015, so it's still very recent, by my colleague Lin Gao, who's just retired from Metabolon very recently. So it's looking at combining whole exome sequencing with metabolomics, global metabolomics approach again, and looking at these genotyped volunteers, they've already been genotyped, who have nominally good health. So what does the genomics tell us, and what does the metabolomics tell us about their health? So I've picked out a couple of examples from that publication in PNAS in 2015, where we're looking at stories about genetic penetrance. Now, individual, this volunteer 3923, the genomics data, the whole exome sequencing data, his physician told him that he had a genetic disease called type 1 xanthinuria, which results in kidney damage and sometimes a rare form of kidney stone. And there's only one kind of treatment for this. You must alter your diet, drink lots of water. So this patient was under the impression, it wasn't really, as you'll find out in a moment, was under the impression he had this disease. Now, when we analyzed the metabolome in the blood, what we found was that the hypoxanthine level was not here to the right, the xanthine level was not here to the right, and the urate level was not here to the left. And I say not to the right to the left, because that's what it should have been if this individual, this volunteer, indeed had type 1 xanthinuria. And to confirm that, the protein was purified and it showed a completely normal function. So even though this person has this rare mutation, they do not have type 1 xanthinuria. So we couldn't really figure this out. It's the result of a single peer-reviewed article and a single individual appearing in that peer-reviewed article being entered into a genomics database. But the metabolomics data clearly refutes the genomics data in this individual. Is there some strange penetrance issue? The second example, B, is of another volunteer lady who did have a genetic disease, but it was not called by the genomics data. This lady had an error in sorbitol degradation. In other words, she had fructose intolerance, which is no laughing matter. It can cause liver and kidney damage. And she had screamingly high levels of sorbitol and high levels of fructose, clearly indicative of malfunctioning fructose bisphosphate aldolase B. So what happened was that looking at the genomics data, it turned out that it was an isoleucine-leucine variation, and it just been called as benign. So the lady was called in, and she was asked questions about her diet. She was shown pictures of cards, with fruits on them, and she said, yeah, I can't tolerate that, I can't tolerate that. So what she needed was some better advice. You must steer clear of this, that, and the other. Otherwise, you risk developing liver and kidney damage. Now, overall, of those 80 people, 20 of them, according to our metabolomics data, had health problems related to a genetic disease, early-stage chronic disease like type 2 diabetes or liver damage, and we found people with drug toxicities related to statins and related to acetaminophen, paracetamol, Tylenol, for example. So the results of this study demonstrate, and this is a direct quote from the paper, that metabolomics could be an effective approach to complement next-generation sequencing for disease risk analysis, disease monitoring, and drug management in our goal toward precision care. So I'm going to speak to you now about that Price et al. Paper and one more, and then I'll conclude, so just 5 more minutes. This was a study of wellness in 108 individuals, and what they did was gather together what they call personal dense dynamic data clouds, enormous amounts of data on these 108 nominally healthy individuals. They collected plasma metabolome, proteome data, genetic traits, and microbiome data, and clinical labs, and as I mentioned already, they looked to correlate these. So what was the paradigm for improving the health of these people? The people were introduced to the concept, and then they arrived, they had the tests taken, then there were multiple coaching sessions based on the biomarkers. Second round of tests taken, have they improved? More coaching. And again, 3 months later, a third round of these tests were taken and more coaching sessions. And at the end of the 9 months, the people were assessed to see if these clinical biomarkers had improved as a surrogate for improved health. What happened was that behavioral coaching did indeed, informed by personal data and coaching, help the patients to improve their clinical biomarkers. This rather beautiful figure you're looking at now is a summary of all the data from those 108 patients from those 3 measurements taken over 9 months of the Spearman interomic cross-sectional correlations between those entire data sets. And you see that there's a lot of correlations in metabolites with clinical labs, and then there's a small number of correlations with proteins and so on. So this uncovered some interesting biology, even though it was only 108 people that were studied. They took polygenic risk scores. They were calculated from GWAS for 127 traits and diseases, and looked for and discovered molecular correlates of polygenic risk. And one example of that was the genetic risk for inflammatory bowel disease, which correlated negatively with plasma levels of cysteine, which is a disulfide form of cysteine. You see the clear correlation. So previously published evidence reported plasma cysteine and cysteine to be abnormally low in patients with IBD. So this suggests a role of oxidative stress in inflammatory bowel disease. I've already mentioned, and I'll mention it again, also some novel associations in terms of biomarkers for cardiometabolic health, and others were identified, such as gamma-glutamyl tyrosine could be a candidate, a kind of master biomarker for diabetes risk independent of BMI. Now, they did this study in 108 individuals, and they're going to go on and do it in 20,000 individuals. And I look forward very much to the kind of fundamental biology, biomarkers, disease biology that could be uncovered by this study. So the key findings were that behavioral coaching informed by personal data helped participants to improve clinical biomarkers, and measurement of personal data clouds over time can improve our understanding of health and disease. including early transitions to disease states. I'm going to introduce you to a second paper published in 2018 in PNAS by Perkins et al. And we'll do a little comparison. So they were doing the same sort of thing, precision medicine screening, but they used whole genome sequencing, global metabolomics clinical laboratory tests as well, but they had a different approach for the rest of the data. They did a diabetic screening test, so it's sort of pre-diabetes, testing if you're insulin resistant. That's something developed by Metabolon. And then the rest of the tests they took were a range of different types of imaging. They didn't apply behavioral coaching. They referred people to a physician if they found a health problem. And I'm just reiterating at the bottom of that slide again why I think metabolomics is of such utility to maintaining health, because the metabolites in clinical laboratory tests correlate well. And in essence, we're measuring hundreds and hundreds, about 1,000 of metabolites in the plasma, which gives you a huge understanding of metabolism and where it's going wrong in health. So the upshot from Perkins et al. Was quite worrying. So in the earlier study with Gow et al., 25% of those normally healthy people seemed to have something unhealthy about them. Now this was a self-selecting group, so it's going to have a bias. But shockingly, 3/4 of those 209 people seemed to have evidence of an age-related chronic disease, such as diabetes or risk of diabetes, atherosclerosis or risk of atherosclerosis, non-alcoholic fatty liver disease, or some evidence of metabolic syndrome. 17 of the study participants needed prompt treatment. 4 of them had a neoplasia, so quite significant. 118 had evidence of diabetes, either full diabetes, type 2 diabetes, pre-diabetes, or insulin resistance from our Metabolon test. 124 had evidence of atherosclerosis or risk. 28 met the definition for non-alcoholic fatty liver disease, that's more than 10%, 1 with NASH. 33 had evidence of metabolic syndrome. And 28 people, so again, more than 10% of everybody in this study had all 4, diabetes or diabetes risk, atherosclerosis or atherosclerosis risk, metabolic syndrome, and non-alcoholic fatty liver disease. And Price's approach, as I said, was to look for improved biomarkers, and they achieved that. So this screening approach found a lot of people with ill health or signs of impending ill health. So in conclusion, with regards to this health approach of maintaining good health rather than treating disease, combining data types such as genomics and metabolomics and other phenotypic measurements assists with health assessment. Healthy individuals are often not. It's hard to say what proportion they are in the population today in the USA, where both these studies were taken, but it seems to be quite high. Analysis of integrated omics data for the healthy assists with disease risk analysis, monitoring, and drug management. And the commonality of the approaches that I've reviewed for you today are genomics, metabolomics, clinical lab tests, patient history, and then some other large, rich phenotypic data sets, such as imaging or proteomics, microbiome, etc. My final word is that metabolomics measures altered phenotype. Almost every factor which impacts the phenotype seems to result in altered metabolite levels. And I don't think that's so hard to understand when you think about the fact that we're all made of cells. And one of the most important things cells do is metabolize. And by producing the most accurate, comprehensive picture of the metabolome, Metabolon believe we can help to uncover causes of cardiometabolic disease and identify biomarkers of the same. And at this point, I'd like to thank you for your attention. If you'd like to write, there's my email address and my colleague Brandon Adams, who helped set up this webinar. Thank you for your attention. I hope you learned something interesting, and I'll be happy to address any questions, or as many as we can in the few minutes remaining. And I'd like to reassure anyone who doesn't get their question answered, we'll write to you with the answer. So the first one is, some of these are technical. What is the typical number of metabolites we might see in a range of different sample types? What kind of samples can we analyze? How much material do we need? Do you need technical replicates? You don't need technical replicates. The kind of variation we observe in our studies is less than 5% in the whole process. So we would always advocate biological replicates. The amount of sample you need, think of the number 100. So if it's a body fluid, 100 microliters. If it's a pellet, 100 microliters. If it's a piece of tissue, 100 milligrams. That's a sort of good general guidance. We can use lesser amounts. For example, we could go down to 25 milligrams, but you'd see a small reduction in the number of metabolites we see. The number of metabolites that we see varies on the sample, but again, as a sort of rule of thumb, think high hundreds. In fecal samples, it's more than 1,000, plus a couple of hundred of those unnamed metabolites. In a plasma sample, it's about 900 or 1,000, plus a couple of hundred of those unnamed metabolites. In a urine sample, we published a paper in 2016 where we were looking at measuring those inborn errors of metabolism in urine, and we identified 1,200 metabolites. In a dried blood spot, 700 metabolites. Exosomes, 500 metabolites, and so it goes. So high hundreds, 1,000, that sort of ballpark is the number of metabolites we might see. And the range of sample types, I've mentioned a few already. Tissues, kidney, liver, heart, muscle, anything goes. Supernatant cell pellets, body fluids, CSF, blood samples, urine. We've looked at skin, sebum, for example. We've had some very exotic samples, breath exhalate, so taking metabolites out of people's breath. I don't know what you call a sample, but when a whale breaches and it sort of snorts out of its nostrils, and there's that mixture of water and, snot that comes out, some biologists can lean over out of their boat and collect that, and we've analyzed that. So I think it's fair to say that we haven't met a sample that you can't get metabolites out of, maybe not rock, but a biological sample should be doable. Someone asks if the gut microbiome, should you analyze feces or plasma? And the short answer to that is either or both. It's not a simple question. Many researchers prefer feces, with the idea, of course, that they're getting their 16S profiling from feces also, so you're sampling from the same sample. But we also see quite a large number of microbial metabolites circulating in the blood. So there isn't a simple answer to that. The answer is, in a perfect world, both. How big is our library of chemical standards? And when I joined Metabolon 2 years ago, it was a little over 4,000, and now it's close to 5,000. So we've added quite a few hundred metabolites. So it's in the ballpark of 5,000. I'll tell you how we make that library. It's quite interesting if you're a mass spec person, but there's a lot more to it than you might think. We have those three separate pieces of evidence, the retention time index, the accurate mass measurement, and the MSMS, which we apply to identifying a metabolite. But what we do when we're making that library is we get that synthetic chemical or biochemical. We run it at 8 different concentrations through our platform, all 4 of those methods, and we're looking to see if there are any effects of concentration. We run it through different matrices like plasma and urine, and we're looking to see if there are any sort of contaminants which elute at the same time, which might have the same mass, the same MSMS, to take away any sort of confounding background chemicals that might interfere. So setting up the library is actually a truly enormous piece of work, and it's probably why a lot of academics can't do it or haven't done it. There's no peer-reviewed article in it, and it will literally take millions of dollars. What are Metabolon's deliverables? What do we provide to our clients? What we provide, I guess you could boil it down to 3 different things. There's this quality control data. I already mentioned that every sample of every batch of samples run, 25% of them are there just for QA, QC. Everything is run on an SOP. There's no variation allowed. I mentioned we like to think of ourselves as being a sort of biology factory, and that's true. You follow the SOP. The only variations are in the R&D department. So we follow strict protocols to ensure a very tiny technical variation, and that when we measure samples today, if we measure the same samples a year from now, we get the same results. So the metabolites are detected in each sample, and their abundance levels are compiled into an Excel file, and I already mentioned that you see the super pathway, the pathway, lots of information, links to external libraries, and then we supply also a heat map. On top of the heat map, we also supply lots of PowerPoint figures showing you the variation between group A, B, C, D, whatever you're analyzing. We do lots of statistics. We'll discuss with you the statistics that need to be done. It isn't necessarily just a PCA. There's lots of other types of statistics, like a random forest, for example, that could be applied to properly qualify and analyze the data. And on top of that, you're able to view that data graphically in our Metabolink client portal in those bubbles of different sizes and colors, for example, I mentioned to you. And you can overlay transcriptomic data and, if you like, at the same time visualize changes in the transcriptome and the metabolome. And lastly, what we deliver is a report. So the biochemist will talk with a statistician and with the client. We'll write a report for you, and it will offer key insights into the biology. This has changed, that has changed, and it means such and such a thing. A nice example from one of those health studies I'd like to point out is we saw 2 individuals. They had altered levels of acetaminophen metabolites, so they seem to be suffering from acetaminophen toxicity. One of them had screamingly high bile acids, one of them had normal bile acids. Then when you looked at the glutathione levels, the person with really high bile acids, which indicates liver damage, had almost no glutathione, but the person who had normal bile acids had really high glutathione levels, which is protective of liver damage, oxidative damage. So if you look at different areas of metabolism, you can understand the answer far better than looking at an individual. So we offer the key insights and a path forward for future experiments. And I think I'm about to run out of time, so I guess I'll hand back to Elizabeth and reassure everyone that we'll collect the questions together. I'll maybe take a day or two, but you'll get the answers back in a written form. Thank you again for attending. I hope very much that you found it interesting and learned something new today.